So the question is, in the medium where E. coli was growing, lactose was added, which induced the Lac Operon. Then why does Lac Operon shut down sometime after addition of lactose in the medium? So before getting to the question, let's understand some basics of this lactose operon. And basically, what is an operon? So operons are what? These are the group of genes, and such genes which have a common promoter. Now when we're talking about the Lac Operon, so this particular operon is studied in E. coli. Why? To understand transport and metabolism of the lactose. And because this pathway is controlled by the lactose, we say the lactose is the inducer of this operon, which means how this pathway will work depends on the presence or the absence of the inducer, which is the lactose. So let's see the first case, that what happens in the absence of the inducer, which means when the lactose is absent. Now here in this operon is present an I gene, and this particular gene works constitutively, which means that it keeps on working all the time. And when this particular gene gets transcribed, it leads to the formation of an mRNA. In this case, the repressor is called repressor mRNA. Then further, translation will occur of this repressor mRNA, and this will lead to the formation of a protein which is called repressor. And as the name suggests, this repressor is going to make the operon inactive. How? This repressor will go and bind to the operator region of the operon, and because the repressor is bound to the operator, the RNA polymerase, which will bind to this particular promoter region, will now have no access to these particular genes. And basically here we see three different genes, the gene Z, gene Y, and gene A. And we know that RNA polymerase is necessary for the transcription, and RNA polymerase will not be able to move here, so we will observe no transcription in this case. And because the transcription will not occur, therefore there will be no synthesis of the protein from these genes. And here the proteins are three different enzymes which have different functions. Now let's consider another situation where the inducer is present, which means that now we have added lactose to the medium. Now again what will happen? This I gene will continue the synthesis of repressor mRNA, because we know that it works constitutively. So we will observe the formation of repressor as usual. But what is different here? That here the lactose, which is our inducer, is present. And what does lactose actually do? This lactose, it will bind to the repressor, making it inactive. And now because the repressor has become inactive, it will not be able to bind to the operator. So what will happen here? Now when the RNA polymerase binds to this particular promoter region. Now it will have the access to these particular genes, and we will observe the transcription of these three genes, which are Z gene, Y gene and A gene. Transcription will occur and the mRNA will form. And further, from the mRNA, after translation, we will observe the synthesis of three different proteins. And what are these three different proteins here? So the protein synthesized from the Z gene is known as beta galactosidase. Same way, the gene synthesized from the Y gene is known as permease, and the gene synthesized from the A gene is called trans-acetylase. And these three different enzymes, they have different functions, and because of that the operon will work and the bacteria will continue its growth. Now if we observe, so first enzyme that is formed here is called the permease enzyme, and this particular enzyme is responsible for making the wall permeable for the lactose. How? It will make the openings in the wall of the bacteria so that the lactose, which is present outside in the medium, can now enter the bacterial cell. And further, the metabolism of lactose occurs, and for that we have another enzyme which is beta galactosidase. So this enzyme is responsible for the further metabolism of lactose. And what happens? It converts the lactose into glucose and the galactose, because basically the bacteria, it cannot use lactose as such. It has to be converted into the usable forms, and the usable forms are the glucose and galactose. So if I tell you one thing, that we have placed the bacteria in a medium where glucose is present and also we have added the lactose. Now in this case, the Lac Operon will not perform its function. Why? Because the glucose, which is the first preference of the bacteria, is now present in the medium, and bacteria will utilize the glucose. Only after the glucose is completely utilized, then the bacteria will switch to lactose and the Lac Operon will start. Now let's see what they have asked in the question. So they have asked that why does Lac Operon shut down sometime after the addition of lactose in the medium? Because we have added lactose into the medium, and that would be in a particular amount, and that would get consumed after some time. And once the lactose is consumed, again what will happen? We will see this particular pathway. The repressor will become active again, because now there is no lactose that can make this repressor inactive. And because the repressor will become active again, it will go and bind to the operator, and hence this whole process of transcription will stop again, and there won't be any synthesis of these particular proteins. So hence our Lac Operon will shut down after some time.